Okay, so hello, good evening. Please let me know if there's any issue with the presentation or my audio at any point. We'll start today's session. Just give me a moment. Okay, so good evening. We are starting off with week 7 of the of our course on genes, genome, and the epigenome. Now, as I had mentioned over the email, we'll be just taking a little bit of a detour from our previous weeks. So till now, we have covered these topics, gene structure, function, and regulation, mutations, polymorphisms, and functional genomics, and also introduction to sequencing methods, where we understood the contrast between Sanger and NGS methods. And specifically, we delved into a lot of detail about the different strategies used in next generation sequencing, sequencing by ligation, sequencing by synthesis, what are the commercial options and how they differ from each other, and what is their relevance, and how you should choose which strategy you should go for depending on your requirement, whether it is short read sequencing, long read, real time long read sequencing, or synthetic long read sequencing. All of these options we had looked at. Then we had begun this particular topic when we discussed the genome, exome, and the transcriptome. And in the last class, we had discussed the entire pipeline of whole exome sequencing. I will be uploading the recordings and the presentation for the same by tomorrow or day after tomorrow. And these are the two topics that I'm skipping temporarily. We'll come back to it after a little bit of a detour because it was getting a little monotonous, the details about sequencing methods and analysis strategies. So today we are going to talk about this particular topic, about introduction to epigenetics and the key concepts in a lot of detail. And while I will be introducing, very briefly introducing DNA methylation and histone modifications, specifically we'll be just touching upon histone modifications today. We'll take up these topics in more detail later on. But today we are going to focus on epigenetics. Now, keep in mind that all of the information, the schematics and everything that I'll be showing today will be from this particular book. This I've just used as a reference. It's a wonderful book by Turner. You can use any reference book that you are comfortable with. And if you are a new somebody who is very new to epigenetics and this entire field, then this is a wonderful book to start with. So you can refer to that. There's also Gardner et al., their book, and a few other commercially available websites, of course, and blogs are also available. So you can look at that as well. But for now, everything you can refer to, that I have referred to and that you can refer to, is from this book. So I've not mentioned the credit in every slide, but this is just some background information. Now, let us come to the topic and why specifically we are talking about epigenetics in the context of this course is, of course, the title refers to the epigenome, but also because gene regulation, one of the baselines on which gene regulation is controlled is via epigenetic regulation. So the context behind that, as we have already discussed before, that while we talk about the central dogma and we talk about the importance of DNA being this stable molecule that carries information, and RNA and its transcription into RNA, and finally translation into protein is what makes the true difference. Control over the genome and control over gene expression is what actually concerns us and what can actually wreak havoc within our system and can completely alter the way our cell functions. So this control, which is the switching on and switching off of genes, not just the switch on or off, but the context in which a particular gene must be switched on and off. Most common example that usually you will find reference for is stress response. So whenever there's a change or a fluctuation in temperature or in the availability of nutrients, and basically any external shift that can be taken care of by the control of genes that would be allowed to be expressed at that point of time, and how quickly the expression can also be sort of retarded and it can be stopped. All of this can be actually understood a lot in a lot more detail. Now, genetic switches are an integral part of most fundamental elements of cellular existence. Of course, that includes cell growth, cell replication, and cell survival. And any anomaly in this network and any disruption in cellular homeostasis gives rise to a variety of diseases. So, this again, just a quick recapitulation of what we already know. The dogma, it involves DNA, which undergoes transcription, a translation, and finally the generation of protein. And the levels of control that we commonly are concerned with or commonly referred to include of course, the replication of DNA that is not shown here in detail, but we are aware of it. But apart from that, a major point of or a major checkpoint over gene regulation is, of course, the binding of RNA polymerase and its ability to actually carry out its transcriptional job or to read the message that is present in DNA and to convert that into protein, initiation of transcription, and the elongation of transcription. Both of these are considered to be regulatory steps or checkpoints for a cell's survival in context of gene regulation. Following that, not just the synthesis of RNA, but rather its correct processing. And by correct processing, we mean that whether or not it's an mRNA or it's a non-coding RNA or whatever is its destiny, based on that, the specific type of processing must take place. When we talk about mRNA, of course, that involves splicing and it involves polyadenylation and capping so that it can be detected or recognized by the physiological machinery responsible for translation, which is the ribosome. And following that, it should be properly transported as well from the interior of the nucleus to the cytoplasm for translation to actually take place. So just synthesis of the RNA is not enough, but gene regulation is also controlled at this step. And of course, here also a large number of proteins coordinate with each other to make it possible. Um, Majorly, that involves proteins which are able to carry out the export of the mRNA. Commonly, they are known as exportins, and they interact with cells. Not cells, they interact with the proteins which are present at the interface of the nucleus, which means that between the interior of the nucleus and the cellular cytoplasm, the membrane that is present, which is the nuclear membrane, there are certain pores present on the nuclear membrane. If you look at the schematic representation of any cell, which you might have studied in your high school or something, you would see that the nucleus is usually represented as sort of like a sponge where it has these little holes. These are called as nuclear pores. And through these pores, the export and import of a large number of molecules, and that also includes mRNA, can also be carried out. And once that control has been executed, within the cytoplasm, the process of translation takes place. Here also, it's not simply the process of translation, but rather multiple steps are involved here as well. First and foremost, the recruitment of the ribosome. It is it's not as simplistic as just ribosomes being present within the cytoplasm and translation just taking place directly. No, correct proteins need correct RNAs need to be detected and recognized by the ribosome. The ribosome can initiate translation at the beginning of the coding sequence, and it can also do so at multiple sites which are present within the RNA, which are called as internal ribosomal entry sites or IRES, and the rate at which translation can take place and whether or not translational control will be smooth or whether it will be hindered, that also depends on a large number of factors. Now, I'm not going into too much detail about it because this can become like an entirely independent course module by itself, the process of transcription and translation. But just for basic understanding, one of the major ways in which control is actually executed at the level of translation is that there are certain secondary structures that the mRNA can adopt. What do we mean by secondary structures? It means that internally the RNA molecule is capable of forming bonds which allow it to assume a hairpin structure or a cruciform-like structure. All of these are very structurally and energetically very expensive for the cell to bypass and support translation instead of just skipping translation. So in such cases, the ribosome can get hindered, and because it's a large macromolecular complex, sometimes it can get stalled, and that can also affect the rate of synthesis and the availability of a particular protein within a cell. So that is the recruitment part. Apart from that, what else can happen? It's not just the ribosome which is controlling the process of translation solely, but instead it is also the availability of tRNA, which is the transfer RNA that will bring the amino acid which corresponds to the codon within your nucleotide sequence that is being read. Again, please refer to our previous discussion on the genetic code and the different types of mutations like frameshift mutations, missense mutations, etc., because that will just brush up your memory real quick. So that is another step at which control is executed. And apart from that, just the availability of the tRNA is not enough. Charging of the tRNA, which means that tRNAs by default, they are simply RNA molecules, right? So the recruitment or the charging of the tRNA means that this tRNA is now bound or is now carrying the amino acid, which will get incorporated within the elongating polypeptide. So that control is also executed by certain enzymes which facilitate the charging process to take place. That is another level of translational control. After all of this has been bypassed, and not just this, the protein sometimes, the polypeptide sometimes also undergoes co-translational protein folding. And if that takes place, well and good. If not, that protein can also be unstable and can also get degraded. So once it has bypassed all of these hurdles, if the final polypeptide is synthesized, its release also needs to be properly executed. And while we don't always talk about ribosomal release or translational release, in a large number of cases, this is actually utilized as a strategy for drug treatment also, like for example, puromycin. So puromycin's a strategy, you can read about it in more detail, but it affects translational release or mRNA release post-translation. So finally, the protein that will be synthesized will be able to do its job. Now, I discuss this in a lot more detail because it's something that we already have studied, but I don't want to get into details about it in this course. But it's something you should know, of course, before we proceed. Excuse. Now, contrary to popular belief, beyond this, there is one more level of gene regulation, which is like along the hierarchy, which is at the topmost level. That is the epigenetic regulation of gene expression. So not just gene regulation, but rather overcoming practical challenges also involves epigenetic regulation and chromatin packaging. These are two things that are so easy to neglect, but so crucial for normal cell survival that we must learn about it. So every nucleated human cell, please refer here, contains around 2 meters of DNA. Right? This is a generic statement. Now, a typical cell nucleus into which this DNA is actually constrained is only about 10 microns, which is 10 to the power minus 6 meters. So imagine the magnitude which needs to be made compatible. So all organisms, whether prokaryotic or eukaryotic, must deal with this problem of packaging a relatively long piece of DNA into a very small space within the cell. That is where chromatin packaging comes in. And not just chromatin packaging, it's not just a static process where you try to smush everything into a very little space, but rather the way in which packaging is executed and the dynamicity of that packaging, which means that it either it can get relaxed and can get tightly wound based on the requirement of the cell at that point of time. That makes it functionally relevant also. It's not just a physical control, it's also a functional control which is executed. So what is the overriding requirement, therefore, for any cell and for its DNA to actually be properly regulated is that the DNA, once it has been um, accommodated, it should be able to actually function. What do we mean by function? Again, first, it must be able to undergo replication with complete accuracy, and the replication should only be carried out once in one round of the cell cycle, and not more than that. The two copies should get separated, which means that there should not be double the copy, and that is also to some extent controlled by the cell itself and by the cell cycle regulator proteins. And of course, the genetic information that is encoded within that DNA sequence should be expressed in a way which is appropriate to that cell at that particular stage and within the environmental conditions that it is experiencing at that point of time. So proper packaging of the chromatin therefore becomes an indispensable step for sorry, for proper cellular function and for cell survival. So taking this, just go back, taking this into consideration, we will now look at the elements which actually allow the packaging of chromatin to be executed smoothly. So this was the context of the problem we are addressing. Now we'll come to the to how that problem is actually solved. So there are two components. First, of course, the DNA, which you want to package, and that DNA should be packaged in a manner that it is accessible when required. And secondly, the helpers that actually enable this packaging and that maintain the sanctity of DNA across the various stages that the cell goes through. So taking this into consideration, we will have to learn about some DNA binding proteins. Why so? Because it is these proteins that are actually able to execute this job. So, excuse me. So how at all was this problem addressed? If you or I, we isolate the nucleus from a sample of cells or from a tissue sample, usually that is a biopsy sample or from any patient-derived tissue, either it is some organ which is cancerous or some organ which has some disease or some atypical behavior when that needs to be studied. What would you do if you were to isolate the nucleus and extract all of the proteins which have come along with the nucleus? Then approximately half of it would be made up by members of one single protein family, which are called the histones. These are the DNA binding proteins which are actually executing the job that we have been discussing all this time. So histones have been known for over 100 years. They were first described and named by Albert Crossell in 1884, and all the members are usually quite small, which is while on one hand, it is a little confusing because they're such tiny proteins. How are they doing their job? Or they're only 10 to 12 kilodaltons. But if you think about it, their small size and their low molecular weight actually helps in better binding and better control over the DNA instead of occupying unnecessary, uh, unnecessary space within the cellular environment. Additionally, one of their major properties is that they're highly basic. They're very rich in lysine and arginine, which are positively charged amino acids that are positively charged at neutral pH or physiological pH inside the cell. One very interesting fact is that these proteins or histones are not found in prokaryotes, although some prokaryotes do have proteins with histone-like structural motifs, but they do not have prokaryotes do not have the exact same protein. They are now universally designated as H1, H2A, H2B, H3, and H4. These are the 5 histone proteins, and among them, you'll learn gradually also. These are the core histone proteins H2A, H2B, H3, and H4. Let us learn about some of their properties next. Okay, please interrupt me and clarify anything if you want it to be discussed, okay? At any point. Uh, yeah, so like I mentioned, among all of the histones, there are some which are considered to be core histones and some which are linker histones. The core histone proteins include H2A, H2B, H3, and H4. Like you can see, their number of residues, which actually dictates their molecular weights, are slightly variable by very few bases. Among them, H4 is the smallest. And Therefore, their molecular mass also varies accordingly. Yeah. And you can see the percentage of residues which actually are constituted by lysines and arginines for each of these types of proteins. And these, as you can see, predominate within the entire cellular population. And therefore, the net positive charge that they carry at physiological pH is also positive and around 15 to 20 degrees in most cases. Um, Linker histone, on the other hand, is much larger, like you can see around 224 amino acids, and the percentage of lysine there is extremely high, around 30% or one third of the protein is made up of lysine itself, which has a functional role to play, which we'll come to later on. But that is pretty much what happens for histones. So, it was observed that when you expose chromatin, Acha, one thing we should be very clear about is that there are certain terms which are often used interchangeably, but you should be very certain that you're using it in the right context. So when we say chromatin, it actually involves both DNA and protein, which are histone proteins, and some NHPs, which are non-histone proteins, all together, which make the genetic material within the cell visible. When I say visible, that's not its job, but that's what makes it visible because it has undergone such extensive packaging that it is now um, visible to the naked eye. Not always the naked eye, but visible using a microscope. And it has actually accommodated a lot of information within a very small space inside the cell. So, like you can see, yeah, like you can see here, when such chromatin is subjected to enzymatic digestion or enzymatic treatment, what it does is that it cleaves the areas in between, like you can see here indicated by the arrows. Sorry, I got muted. Yeah. So, like you can see, there are multiple sites where nucleus digestion can take place, and as a result of that, what you'll get are these digested chromatin fragments. Once you can subject these sequences to the removal of the bound proteins, usually by protease treatments. What you'll eventually end up with is the DNA, which has been protected because of the binding of all of these proteins. And you can actually visualize it by running it on a gel, where you'll get many fragments of many different sizes and sometimes smears as well, depending upon how digestion has taken place and how much of the region was accessible to the enzyme. So interestingly, the same pattern of DNA cleavage could also be achieved in nuclei that lacked endogenous nuclease by incubating them with a bacterial enzyme, micrococcal nuclease, under conditions that allowed it to do its job. Most nuclear DNA was susceptible to this very specific pattern of cleavage, showing that the repeat packaging unit, whatever it is, when I say whatever it is, it is because initially the nucleosome was not known. Now we understand it. But what this initial packaging unit, which was not clearly discovered, was expected to do was that it should package most of the nuclear DNA rather than just a minor fraction. And yeah, so that was pretty much what was understood from this point, that the DNA is present in this balls on a string fashion, and therefore the string portions can actually be digested by enzymes like MNAs, while the rest of it remains undisturbed. Now let us understand this concept of genome packaging in a lot more detail. As I previously mentioned, beads on a string or balls on a string. Yeah, this appearance that you can see, this is an electron micrograph of cells that have actually been visualized. I think it was some sort of a blood cell. I cannot recollect it at the moment. Again, this is taken from the book, so you can refer to that resource also. But as you can see, the scale bar here is of 100 nanometers, which is quite large by uh, when you compare it to other molecular structures. And this appearance that you can see, it looks like they are little beads that are present bound to a string. And this appearance actually suggested that there are small units of chromatin packaging, and these units are what we understand today as nucleosomes. And additionally, on the left, you also have a quick recap of the way in which DNA is arranged. I think we had discussed this also initially. Um, but basically, you can see that there are two grooves. There is one major groove and there's one minor groove because of the way in which the DNA double helix turns. There is a periodicity within the sequence which allows for turns to repeat themselves at certain distances, approximately around 3.4 to 3.5 nanometers, and that accommodates 10.5 base pairs. Again, this is a math, between 10 to 11 base pairs can be accommodated. So overall, this is the linear structure of DNA on the left. And after packaging, what is actually visible to the microscope is something like this on the right, which is the beads on a string structure. Now, to understand and to interpret the actual need for this sort of extensive genome packaging is this fact, where you can see that different types of bacteria, whether prokaryote or eukaryote, depending upon how evolved they are, a lot of parameters are very different. Like, for example, you can see the comparison here between E. coli, Saccharomyces cerevisiae, Sinorhabditis elegans, Drosophila, and humans, you can see that the amount of DNA or the megabases of DNA is quite variable, with the highest being present in humans. And the number of genes which are encoded from this chromatin sequence is also extremely immense. And also, it is necessary because these organisms are a lot more complex, and therefore their survival is dependent on a large number of things. And the density of genes also can be seen to be different, but if you look closely, you'll see that there is a difference in the pattern which is present, that while number of coding genes or the gene density is quite variable, it is the lowest in humans, and that suggests that most of the genome is actually not protein coding, and therefore the gene density is not very high. So additionally, also what is understood here is that the organism sometimes, for example, here Drosophila, which has around 120 megabases of DNA, that has 60 megabases of largely repetitive non-coding DNA, which is called as the heterochromatic DNA or heterochromatin. That is not included here, and it is is found in humans and other mammals, and therefore it's just another point of variation. Now, practically speaking, for example, you are in the lab and all these discoveries are something that you can see, the discoveries that you read about today were initially the conclusion of some sort of an experiment, right? So how was this done? To study the arrangement or the packaging of the chromosome, chromatin fragments were separated into different units of discrete size. So for that, you prepare chromatin and you layer it on top of a sucrose solution in a centrifuge tube, and then spin it at very high speeds. As a consequence, the chromatin fragments will move down at different speeds. And after centrifugation, what you expect to see is that the larger or the largest proportion or largest part of the chromatin preparation will actually be able to settle down. And after a few hours, you'll see that discrete bands of chromatin will separate within the sucrose solution. And if the DNA from each peak fraction gets resolved by electrophoresis, so yeah, so you take little fractions either from the top or the bottom. How do you do it? You have this, for example, you have a tube which is filled with some sort of a sucrose solution, and you add your chromatin preparation into it, and then you centrifuge it. Then you keep taking subsections from this particular sucrose gradient or sucrose solution, and you can then observe their migration or their electrophoretic mobility. The chromatin bands can actually be detected by separating the solution in the tube into very small fractions, like I already mentioned, and measuring their optical density. if the DNA from each of these peak fractions is resolved by electrophoresis, it will form a single band of discrete size, which here, um, pretty much that only. Um, Yeah, the chromatin bands can actually be detected using these small fractions, and their optical density can be measured. And what you expect is that the DNA should be able to form a single band of a discrete size when ETBR staining is carried out. Protein electrophoresis shows that each fraction contains the same set of four core histones in equimolar amounts. And here you can see Kumasi blue stain, and H4 binds the dye less well than other three histones, so it stains less effectively, but it's still usable. So basically what we're trying to say is that if you have a mixture of the end um, or a representative material of the chromatin which is present within one cell or a population of cells, and you separate it using density based centrifugation methods, then you will get these different populations which will represent the different types of histones which are present. Now let us understand this in more detail. Just a second, just give me one moment, please. Okay, yeah, sorry for the interruption. So we will learn about the nucleosome in more detail. Again, let me just drop another reminder. If you have any doubt at any point, please interrupt me and please ask me whatever queries you have. So we were talking about the genome packaging, and now we are discussing the nucleosome. Why did we shift from there to here? When you saw this image about beads on a string, what you see as these tiny little unit is what represents one nucleosome. Therefore, the most easily discernible unit of the entire genome packaging or the structural packaging image that you can see here is actually the nucleosome. What does it exactly mean? This is what it means. It means it is the unit of packaging of the chromatin in an eukaryotic organism. So how do we understand it experimentally? If nuclei, again, in continuation with the experiment from the previous slide, if nuclei are incubated with micrococcal nuclease or any such nuclease enzyme, and then the chromatin is isolated under mild conditions, it can actually be separated by centrifugation on sucrose density gradients into a series of fractions of increasing size. The peaks on the gradient are particularly clear if histone H1 is dissociated from the chromatin by exposure to moderate salt concentrations. Why do you want to do that? That is because histone H4 size is quite different, and it sort of skews the results for all the other histones. The peak fractions isolated from sucrose gradients under equimolar amounts of H2A, H2B, H3, and H4, and the size of the DNA in each fraction corresponds exactly to that of the individual bands in the DNA ladder generated by digestion of nuclei with either endogenous or added nucleases. What do we mean by all of that? First of all, I hope you have context of sucrose density gradient. If not, please let me know. But briefly speaking, what exactly you do is that suppose you have, let me just switch on the video. I think that'll, yeah. So what I'm trying to say is that if you usually you would have like a long beaker or flask or some sort of a test tube or a vessel basically. What you'll do is you will have sucrose solutions which are of very different percentages or different densities, because sucrose consider it like sugar solution, for example. Depending on the amount of sugar that you dissolve, you will see that there's a difference in the density. So what you can do, just hypothetically speaking, if you have 10 different percentage of sugar dissolved in 10 different types of solutions, or just a different percentage of sucrose in water, it will give you a range of sucrose solutions, right? What you can do is you can layer these one on top of the other and then subject it to centrifugation. Based on its density, anything that you add to this density gradient can also separate according to the buoyancy or according to the density of the particle that you're also using for this separation purpose. So for example, here, if you are talking about the chromatin, or if you are talking about histones, for example, then in that case, depending upon what is the density of the histone proteins, they can also get segregated across the gradient. Now, what we are trying to say here is that after this separation has been done, you can separately isolate each of these um, density regions. And how that can usually how that is done is either you use some sort of a stopper, or you can just isolate that layer separately. For example, if you have studied um, ultracentrifugation, where sequential ultracentrifugation is done to separate different parts of the cell or different cellular organelles. So you take the entire cell lysate and then you can subject it to increasing speeds so that certain densities get precipitated while others don't, and then you can remove the rest of it and you can segregate out one particular fraction. So when these peak fractions of sucrose density gradients are actually studied, it can reveal that you would have different types of histones like H2A, H2B, H3, and H4. And the DNA that you get from each of these fractions, it will correspond to the DNA ladder that you get when you digest nuclei. The DNA ladder, I think we showed already here. Yeah. So this is the DNA ladder that you get. By ladder, what we mean is that for one particular peak, say for example, consider histone H3, H4, H2A, H2B. Each of them bind to certain regions of the genome, and therefore wherever they bind, they protect that region. So if you take the solution which has histone H1 bound DNA, and you take the percentage, or you take the histone H1 isolated from that same section, what you'll see is a concordance between the two, which will be a confirmation of the fact that yes, these regions of DNA were actually protected by H1 binding, and it will confirm your original hypothesis as well. So DNA cleavage and ultracentrifugation of chromatin preparation are the key experiments that were performed initially, and that actually were able to hint towards the fact that epigenetic regulation and chromatin packaging actually have a very dynamic role to play in cell survival and gene expression regulation. So effectively, based on whatever was understood at that point, one of the major contributors to this particular field was Roger Kornberg. What Kornberg actually suggested, these are some of the key hypotheses that were suggested by Kornberg. First, chromatin consists of a fundamental repeating unit, which is made up of nearly 200 bases of DNA. Usually it is expected to be somewhere around 160 to 170 base pairs in this range. And it also has these histone proteins along with it, H2A, H2B, H3, and H4. These histones actually form an octameric structure. So those four, I don't think I have that figure here, but basically think of it like a small grenade of some sort, which is made up of these individual subunits. And around it, again, we'll come to this later, around it, you will have DNA wrapped, which will actually constitute an entire nucleosome. So this nucleosome is then also undergoing multiple levels of packaging. I think we had discussed this in the initial class, and it is actually the figure which is present here in your course handout as well. If you look here, these individual structures are the nucleosomes. Yeah, that's actually a good figure. Um, if it is interesting, you can go and look at this in better clarity. But basically, what you see here are individual nucleosomes, and then they undergo multiple levels of packaging, which forms a thicker sort of like a strand, a 30 nanometer fiber or chromatin fibers, which undergo multiple rounds of packaging that eventually end up forming the chromosome that you're able to see, that protects the DNA, that maintains the ability of the DNA to undergo proper replication only once, so that there are no mishaps. And the two copies should undergo proper segregation between two daughter cells, and the DNA should be dynamic such that it can get expressed when it is required. Yeah. So one chromatin fiber, like I just mentioned, one chromatin fiber, sorry, undergoes packaging such that it consists of many such nucleosomes forming a flexibly jointed chain. This corresponds to the beads on a string fiber observed under electron microscope. Analysis of histone-histone interactions. Again, these are all experimentally validated. I've mentioned some experiments. I had to skip some because if you think about it, all these discoveries, there are a number of experiments that have actually supported each of these hypotheses. But basically what you see here are that 8 histones are all connected into a single unit, which is called as the histone octamer. And this unit, it's not continuous. They are all independently folding subunits, which can be separated or which can be dissociated. And that can be done by the manipulation of ionic conditions such that it forms a H3H4 tetramer. That means two units, one H3H4, another H3H4 together, coming together and forming one tetramer. So it is not that there are two H2A, two H2B, two H3A, and two H3 and two H4. It's not like that. H3H4 forms individual not dimers but heterodimers, and then they come together to form a tetramer. And apart from that, H2A, H2B dimers join in, and all together an octamer is formed. So 8 separate histone proteins form the core histone. Um, Furthermore, the first experimental clues, they came in, and again from nucleus digestion studies, when a careful analysis was carried out about how nucleosomal DNA was actually digested by the endonuclease DNase 1, it led to the proposal that DNA actually is coiled around the outside of the histone octamer. We'll come to this in more detail now. So what is the objective? The objective is to understand the nucleosome in extensive detail. What do we first know? We know that the core is composed of multiple protein subunits, not even subunits, but multiple proteins coming together to form a multimeric subunit, which are all part of the histone family. There are different proteins that are interacting in a manner such that it is eventually forming the octamer, but it is not the simple association of 8 proteins. They are forming individual subunits, which are finally all coming together. And next, the next part of it says that around the octamer, the DNA is actually wound on the outside. It's not that like, again, this is an example I give almost every time in every course that I teach epigenetics in. So this is think of this mouse that you might have as your as the core, as the histone octamer core, and around it on the outside, DNA is wound in this manner, right? The other option, which is why experimental validation is necessary, is that if your DNA was on the interior and it was forming sort of a loop, this is getting hazy somehow, but if this was forming a loop and the presence of the octamer covered it, then that would also protect that DNA from DNA digestion. So that would have been another possibility in which this packaging could have been done. But the fact that packaging is on the outside and DNA is accessible allows this packaging to be a lot more dynamic. Instead of simply being a protective step, it is actually a gene regulation step. So the way in which this packaging occurs also, of course, has been the result of multiple centuries of evolution, which has contributed to the genome architecture in the long run. So yeah, so this was also understood by certain types of certain studies, and the study that I'm referring to here is the way in which the enzyme DNase 1 digests. So we'll come to it later, but why understanding the fact that the enzyme which you use also makes a difference in the way in which DNA gets digested is because of the mechanism that is opted for by the enzyme to do this digestion step. So in context of the enzyme which we are studying, two enzymes are mostly or were mostly used whenever we talk about epigenetic studies. One was MNase, which is micrococcal nuclease. The other is DNase, which is DNase 1, which is another sort of an enzyme that is able to cut DNA. The micrococcal nuclease works in a very specific manner. It sort of acts like bolt cutters. If you have seen the sort of cutters which are able to pierce through cables and other wires. So what they do is, this is the central nucleosome. Like you can see the figure here, it's a better representation. And what they do is the enzyme needs to straddle the DNA, and only then it is able to cut it. Because of this reason, the enzyme is only able to cut DNA that is resting on a surface. Therefore, nucleosomal DNA is protected from cutting by MNase even though it is located on the outside of the histone octamer. Now, this is a heavy statement and a very informative statement. What do we mean by that? Please understand that that there is another set of terminologies that we have not referred to directly here, but there are some terms which you must have heard of. One is euchromatin, another is heterochromatin. Euchromatin is completely exposed chromatin. Heterochromatin is that which is currently packaged and is inaccessible and is part of the chromatin fiber which is present within a cell. Apart from that, when you look at packaged chromatin, within that also, these regions which are wound around the histone octamer, they are called as nucleosomal DNA because it is actually helping the formation, or it is a part of the nucleosome. A nucleosome is only formed when there is DNA plus there are proteins. So this particular DNA is nucleosomal DNA, and what is present outside of it is linker DNA or is accessible DNA that is present between two nucleosomes. Why are we stressing on this? That is because even though this DNA is present on the outside and is exposed and is allowing transcription, translation, um, gene regulation, modifications, all of this to take place, enzymes like micrococcal nuclease are still not able to cut in that region. And that is the reason why we were able to see that DNA ladder that we saw a couple of slides ago. That is because the enzyme sits in a way that it needs to completely encompass DNA, it needs to straddle DNA, and then it is able to actually cut it so that in those regions you will have some double-stranded breaks. That is why this entire region, which is actually present around the core histone octamer, is protected, whereas regions surrounding it undergo digestion, and therefore you get information about the nucleosomal DNA. In sharp contrast, the way in which the DNase 1 enzyme works is quite different. And in this case, what you see that it attaches to the DNA that it wherever it attaches to in the regions or the locus where DNase 1 actually binds. What it does is it makes nicks, or it makes single-stranded cuts, or it can make double-stranded cuts in the DNA double helix. When the DNA is laid along a surface, DNase 1 can still make these strands, but only the DNA farthest from the surface can be cut easily. So here you can see that DNase 1 has a different pattern. What does it do? It cuts nucleosomal DNA at sites which are farthest from the histone core. So therefore, one major difference between the initial and between MNase and DNase 1 enzymes is that DNase 1 can preferentially or does preferentially cut this nucleosomal DNA. So while this entire region, which is present in close association with the histone core and forming the nucleosomal DNA, is inaccessible to micrococcal nuclease, the same region is actually accessible to DNase 1. Because of this preferential single strand cutting at the sites indicated by arrows, which are majorly these regions, like you can see here, the most frequent pieces of DNA generated by DNase 1 treatment of nucleosomes can actually differ in size by around 10 base pairs, that is every turn of the DNA double helix. This periodicity can actually be detected by electrophoresis, and that will indicate how long that DNA actually is. So what is the key difference? First, the DNA that it can actually bind to and actually chop is different between MNase and DNase 1. The way in which the enzyme sits on the DNA is quite different, which changes the accessibility of the DNA of the enzyme to the substrate also. When DNA is laid along a surface, then in that case, DNase 1 can still make these nicks, but only the DNA which is farthest from the surface can be easily cut. So therefore, that actually allows us to distinguish between different regions of the genome. It also shows us how the shift in the position of the nucleosome, like if you imagine a straight piece of DNA and you imagine a nucleosome bang in the center of it. This is just another hypothetical situation. If it is at the center of it, then it is not, since it is not static, after some time you can expect the nucleosome to shift a little bit in either direction. Why is this necessary? This is actually a process known as nucleosomal breathing. What does it mean? It means that the region where the nucleosome is actually formed, that is where the histone is binding to the DNA sequence of interest, is not completely constant. There are a few bases around, I think it was 20 to 30 bases on each end, where the nucleosome moves a little bit to adjust for transcription and translation and based on the requirement. And it's because it is a dynamic process. It's not that there is a um, there's like a very strict checkpoint, but there is a possibility that some regions will have very strict boundaries for how much of the nucleosome can actually move. That is actually moderated by the linker histone, which is histone H1, that occupies a position outside the core octamer, close to where the DNA strand enters and exits the nucleosome. This is consistent with its effect on nucleus digestion and with the observed stoichiometry of one H1 molecule per nucleosome. A nucleosome complexed with H1 histone sometimes is referred to as a chromatosome. What did we say? A lot of things were said. First of all, if you remember, when we first introduced histones, we saw that there are 5 different subtypes that are involved in chromatin packaging, H2A, H2B, H3, and H4. Apart from that, there is also a linker histone H1, which is not a part of the core histone octamer, but instead it sort of acts as a stopper. The stopper that I just mentioned, that job is often fulfilled by the linker histone H1. Sorry. So what does it do? It actually, after the completion of that nucleosome, it sort of stops or it defines the end of the region which will be a part of the nucleosomal DNA. Then there is a linker, then there is another nucleosome, and so on and so forth. This is also something which has been observed in nucleus digestion and with the stoichiometry of one H1 molecule per nucleosome. What does it mean? It means that within the histone octamer, while you will have, say for example, within a cell, if there are n number of nucleosomes, then within n number of nucleosomes, there will be 2n number of each of the core histone octamers, right? Because in each octamer, you will have 2H3, 2H4, 2H2A, and 2H2B, but you will have only one H1 which will be present, and that will be present on the outside. So this stoichiometry, that for every one histone H1 molecule, there will be 2 H3A, H3B, and so on and so forth. This entire 3D structure, which is the nucleosome, plus H1 is altogether called as a chromatosome. So that's what you see here, the dimensions of the nucleosome core particle and possible location of linker H1 histone in the chromatosome. So this complete structure is a chromatosome. This is the nucleosome core particle. Please have these definitions very clear because these cannot be used interchangeably. It is with and without and very important protein, which is the linker histone H1. Also, here you can appreciate the dimension of the of the nucleosome and chromatosome as well, where you'll have around 55 Armstrong height or depth, and you'll have around 110 Armstrong of the width, almost double of it, or exactly double of it. And another term you see here is the dyad axis. Dyad axis is the axis of symmetry. That means if you cut the nucleosome by half here, you will see exact symmetry at the top and at the bottom, which is also indicative of the arrangement that the histone molecules assume within the entire nucleosome. So yeah, if there are any doubts till now, please let me know and then we can proceed forward.
It is clear, ma'am.
Yeah. Okay. So now let us understand the details of these individual proteins in a little more detail. Uh, Here you see a term called as a histone handshake, which is again indicative of the 3D structure, which is assumed by these histone proteins and how they interact with each other. What you see as a commonality in the structural organization of these proteins is the presence of a very unique and very characteristic fold, which is called as the histone fold. What is it? The histone fold is the presence of certain of alpha helices basically in a certain orientation that is present in all of the histone proteins. The central globular domains of all the four core histones have a set of three alpha helical regions that form a characteristic structure known as the histone fold. So this is alpha 1, 2, and 3. This is linker 1 and linker 2. The shaded boxes labeled A1, A2, and A3 in the upper part of the figure represent the alpha helical regions. L1 and L2 are the non-helical loops between these helices. The broken lines, they represent alpha helical regions that are not forming a part of the histone fold. So it's not present in all of the proteins as well. In H3 and in H2B, you can see that these are present. The fold domains of H3 and H4, they interact in the core particle. The H3, this is H3, this is H4, their fold domains actually interact in the core particle to form the histone handshake, which is shown here. The fold domains of H2A and H2B also interact in the same way. Why is this important? It is important because this shows you the assembly of the histone octamer, how that takes place. Because think of it from a bird's eye view. Within a cell, we are talking about the entire rearrangement of the chromosome and how important it is, and it's lots and lots of things. But if you think about it, these are also simple proteins, right? Histones are also proteins. They will be synthesized by the cell and they will undergo folding. But their arrangement and their functionality will be determined by all of these different variables. One of those variables is the way in which these proteins interact with each other. Two of those key interactions include first, the fold domain of H3 and H4. They are interacting with each other. Like you can see here, this is H3 and this is H4. They are interacting with each other in sort of like this fashion, which is the histone handshake. The same is seen for H2A and H2B also. And these individual heterodimers can then interact with each other to form tetramers and eventually form the octamer. In the core In the core octamer, the three helical regions in each of the four histones are arranged in a very similar way to give the tertiary structure known as the histone fold. These, yeah, this histone folds of H3 and H4 and H2 and H2B, they also interlock with each other to form the histone handshake. So overall, the 3D organization of all of these histone proteins via their histone fold regions, which includes 3 alpha helices connected by linker regions, altogether forms the histone handshake. It is this handshake interaction that gives rise to the stable H3, H4 and H2A, H2B dimers that are fundamental in the core octamer that defines the histone core. Following this, yeah. Oh, I think I kept that much information only. Yeah. But overall, that is the histone handshake that forms the entire core octamer around which the DNA is wound. But that is not all that is present, because it is not a simple globular protein that is like, it's not a simple sphere. It's a protein. It's a polypeptide chain that has undergone folding in a manner to assume this globular sort of structure, but it is not a completely spherical like a spherical structure, right? It actually has extended regions, which you can see here. It's again another schematic representation. These are all of these are, of course, schematic representation, which is for ease of understanding. A better way to actually interpret it is to, like if you have used Chimera or Pymol or any of these softwares, you would have seen that there are different ways of representing protein secondary and tertiary structures. So that involves many a times using the backbone of the protein and using different sorts of projections like the ribbon diagram or space filling diagram, and you can use all of those to understand how it actually looks in real life, or very close to what it actually looks inside the cell in real life. So what we are focusing on right now is the fact that because these are sort of extensions of the protein, there are certain regions which are not directly a part of the nucleosome, but they are components of the nucleosomal structure, and they are called as the histone tails. Amino acid sequences of the N-terminal tail domains of the 4 core histones. So why again the N-terminal domains? Because they usually project out of the nucleosomal core, and you can see that these are the different okay, another thing I just remembered it. If you look here, if you look at this particular diagram, you'll see the contrast or the slight differences also between each of these different histones, but the length or the amino acid number is variable between different histones, among which H3 is the longest histone molecule or the heaviest histone molecule, while H4 is the shortest, and H2A, H2B are quite close. So these are also some subtle differences. Again, not a very huge impact, but that is how they are, and you should be aware of the differences between histones as well. So yeah, another reason why it was actually important is because based on how long that particular amino acid sequence is, you can also understand how long the tail actually is. And the significance of having a longer tail on that particular histone is the fact that the longer the tail, the more probable that there can be modifications, or there can be some changes or some alterations, or the more it can actually interact with something else and affect the way in which chromatin packaging is regulated locally in that region. So here you see the amino acid sequence of the N-terminal domains of the 4 core histones. Residue numbers are given above each sequence, and positively charged residues, which are lysine and arginine in this case, are indicated below. The vertical arrows indicate the sites which are farthest from the N-terminus at which the protease trypsin can cut the tails in the chromatid. These sites define the extent of the exposed N-terminal domains. What does this mean? This shows you here the vertical arrows here. They indicate the sites which are farthest from the N-terminus where trypsin can actually cut chromatin tails. So that is how far N-terminal domain tails are actually exposed. So what you see here is not the end of the protein chain or the C-terminal end of the protein chain, it's the N-terminal end. So regions which are exposed are regions that can be cut by enzymes. Again, the same logic, anything which is open and accessible can be cut by enzymes. So in this case, because it's a protein, using endonucleases will not do the job. So you use a protease. In this case, the protease that has been referred to is trypsin. So the site till which trypsin cuts is, again, you can see it's different in different histone proteins, but that defines the length of the N-terminal tail. And that you can see here is also the longest for H3, and is also quite extensively positively charged. These plus signs indicate the charge of the amino acids which are exposed as part of the tail. So, yeah, so sites which are located further towards the C-terminal end get protected from cutting by the core DNA. So because it actually forms a part of the nucleosomal core, that region is not cut by trypsin, the rest of it is. Underlined regions have not yet, again, please keep in mind these are all excerpts from a relatively older version of the book. You can keep yourself updated with the current version as of now. But at that point of time, underlined regions which are shown here, here, and here, they were not located by X-ray analysis of the nucleosome core and might be unstructured regions which cannot be um, which cannot be captured in an X-ray crystallography experiment, whose crystal cannot be made because it is that dynamic a protein. But I'm sure a lot of progress must have occurred on this by now, where people use NMR for such soluble proteins which are difficult to crystallize. And also on the basis of their amino acid sequence, some regions in H3 and H4 have a high probability of adopting an alpha helical structure, and box which are shaded here, some regions are shaded here, they take up that alpha helical sequence like we discussed, the histone fold. And box regions are short stretches of known alpha helix, and box region in H3 is the stretch of amino acids that actually traverses the core DNA. The residues at the N-terminus region of H2B are missing. Yeah, this is not important. I think I copied it from the book, so it has come here. But overall, what we see are different properties. First, the length of the histone tail is different for each of the different histones. Second, the presence of positively charged amino acid is also variable across different histones. Third, there are certain regions which form a part of the helix that constitutes the histone fold, which performs histone handshake and constructs the entire central histone octamer. Sorry. Fourth, you can see that certain regions are very dynamic, and those are very easy to cleave by trypsin because the enzyme is able to access it, and B, its structure is difficult to crystallize, so a little bit of uncertainty also remains there. And yeah, these are the various properties or the various key characteristic features of the histone tails. So this is the diagram that was proposed based on the information that was available at that point of time. The nucleosomal core particle and the histone tail domains. The diagram is loosely based on the X-ray structure of core particle crystals. Complete turns of the DNA helix are numbered from the diad axis 1 to 7 in one direction and -1 to -7 in the other. So you can see here you can see -1, -2, -3, and so forth. And here you can see 4, 5, 6, and 7. So this is the orientation of the DNA. It's just based on the axis and based on how symmetric they are with each other. Both the N-terminal tails of H3 and H4 are shown. And yeah, uh, the tails of both are shown, and just one for H2A and H2B have been shown here. So these are the H2B and H2A tails that have been shown, and for H3 and H4 both have been shown. Um, the sites at which the tails emerge from the core DNA are based on crystallographic analysis. The length of each tail is defined by the position of the most distal trypsin cutting. That means the length, the farthest that trypsin is able to actually cut. That will define the length of each of these tails, and it assumes that each tail is fully extended at 3.7 angstrom per amino acid. In fact, tails are very likely to adopt defined structures in vivo, if only through interactions with other proteins or with core DNA. Stippled regions of core DNA become more sensitive to nucleases after removal of histone tails. So once histone tails are removed, these regions of core DNA become more susceptible to nucleases. So overall, it's a summary of what we have already discussed and the orientation that they assume. I think we have only, yeah, two more slides. We'll wrap up soon. We'll wrap up in like 10, 15, 20 minutes. So, yeah. So, what have we discussed? First, the structure of the nucleosome, what makes it up, what are the components, what are the properties of those components, what is the structure of each of those components, that is the structure of the histone, how histone proteins interact among themselves, um, what is their job also static? Are they only, serving as a scaffold or um as a base on which DNA can be sort of immobilized, and what is the job of the tails that project out of it. So these are some of the common things that we have already discussed. Now, apart from what we already know, histones also undergo a large number of different modifications that have a very, very major role to play, and we'll learn more about this in context of the sequencing techniques that can be used, and how they are extremely useful in today's time and have been useful and contributed to academia and research for a very long period of time. So, amino acids that are present within the N-terminal tails of histone proteins are capable of undergoing modification by certain specific enzymes in vivo. What are the different types of modifications? Most commonly, you'll find two, which are acetylation, which is the addition or removal of the acetyl group, and the second one is methylation, the addition or the removal of a methyl group, which is a CH3 group. Apart from that, you also have phosphorylation often occurring, and you have ADP ribosylation. Sometimes, very rarely, you also have the addition of ubiquitin and sumo group. All of these are possible modifications that take place, but among these, again, as I mentioned, the addition of the acetylation and the methylation of DNA are marks that have a lot more significant impact. You'll find it not only more widespread within the genome, but also more relevant when we are trying to carry out any research study. So, what exactly happens there? You have you have the like look here. You'll see that this is your protein of interest, to which, when there is the action of a histone acetyl transferase or a HAT enzyme, what it does is that it uses acetyl coenzyme A as the donor, and it transfers the acetyl group onto your protein of interest such that it now carries this COCH3 group along with the at the N-terminal tail along with the NH, and now it has become an acetylated protein which carries a negative charge. Here, this bitter form is shown, or the physiologically stable form is shown, but COO minus group carries a negative charge. And in contrast, because this is a reversible process, the same protein can undergo deacetylation, which is the removal of an acetyl group by HDACs and HDAs, which are able to remove the same. Examples like butyrate, TSA, all of these help. And what it does is it gets rid of this group and you have your initial protein. Now, why is this necessary? We'll come to it. Histones in general, okay, let me come to it later, but just think of it in this manner. Histones in general, they carry a very high positive charge because you have those arginine, lysine groups, etc., right? Why that is important is because the backbone of DNA, the phosphodiester backbone, that carries a negative charge. To facilitate very strong interactions between the DNA and the histone core octamer, the positive negative charge interaction helps by leaps and bounds. It is one of the major stabilizing forces between these two entities, the protein and the DNA. Now, when there is the addition of an acetyl group, sorry, when there's the addition of an acetyl group, what it does is it disrupts this interaction between the DNA and the protein, because now the protein is also carrying a negative charge. As a result, the strength with which, or the intensity with which DNA and protein could have interacted with each other don't do so anymore. And as a result, DNA becomes loosely wound, DNA becomes a lot more accessible, and therefore transcription goes up. So, what acetylation is commonly known as is an activation mark, because it is activating transcription, or it is helping transcription, and it is opening up DNA to be read by the transcriptional machinery. The exact opposite is usually, again, please keep in mind that these are not very absolute changes in DNA protein interaction and gene expression. These are usual ideas. Methylation, in contrast, is expected to do the exact opposite, and methylation is often understood to be a repressive mark, but again, there are exceptions to this. We'll come to it. Here you can see that the acetylated isoforms of histone H4 can also be resolved by electrophoresis. So like I mentioned previously, histones carry a net positive charge, a very high net positive charge. And if it is subjected to electrophoresis at low pH, what they do is they move towards the cathode, the positively charged electrode. For each lysine that undergoes acetylation, the net positive charge gets reduced by 1. As a result, the rate at which the histone moves towards the positively charged electrode becomes slower. The acetylation, or the acetylated isoforms of H4 separate particularly well, and isoforms with 1, 2, 3, or 4 acetate groups can be clearly resolved on gels. So basically, every time there is an acetyl group that is added, it is reducing the net positive charge and the ability of that molecule to move towards the positively charged electrode. And as a result, you will be able to see more number of bands here. So yeah, so more the deacetylase also. Deacetylase inhibitor means that it is inhibiting the enzyme which is able to remove acetyl groups. As a result, more and more acetyl groups will keep on getting added, and therefore you'll see a shift there as well. Um, yeah, so acetylated forms of H4 separate very well, and you can see it clearly on gels containing high concentration of acetic acid, urea, and detergent Triton X100. More acetylated forms are very rare in untreated cells, but if you expose cells to histone deacetylase inhibitors, you'll have more and more acetylation, and you'll have more and more um, you'll be able to see these intermediate forms a lot better, and migration towards the positive electrode will also reduce. This is the anode, this is the cathode. So from here to here, this movement will actually get retarded. This is the movement towards the cathode that will get reduced by a lot. Um, yeah. So then the second type of modification, which is methylation, does a relatively different, or rather almost the opposite effect on the histone and on transcriptional activation as well. Usually it is considered to be a repressive mark, in contrast to what acetylation is expected to do. The first sequencing studies of histones H3 and H4 showed that certain lysine residues undergo methylation on their side chain epsilon amino groups. Methyl lysine, like acetyl lysine, is also chemically stable. This modification is also complicated by the fact that 1, 2, or 3 methyl groups can be added to each epsilon amino group. What does this mean? It means that for the same histone at the same site on the same histone, either 1, 2, or 3 methyl groups, because methyl is a small group, each of these can be added. And depending upon what sort of a um, activation or a repressive mark is added, the effect of it can be different. Like sometimes H3K4 trimethylation is considered to be, not sometimes, but always it is considered to be an activation mark, while the addition of a single methyl group is repressive in nature. So it is very context specific, and it is also dependent on where that methyl group is getting added. And the high energy compound S-adenosyl methionine, or SAM, acts as the donor, the methyl donor in this process, um, for the methylation mark to actually be added onto D or onto histones. So yeah, this is monomethylated lysine. This is S-adenosyl methionine, from which the CH3 group is getting transferred by the methyl transferase. Here, I would quickly like to remind you of something. Remember DNA methylation. This is the exact figure we had discussed, I think, in the second class, most probably when we had just started the course. And why I'm bringing this back is because I want you to be very alert and not confuse histone methylation and DNA methylation. DNA methylation is completely different. Sometimes it is the influence of both of these molecules, and the type of modification which is occurring in both of these molecules, and the effect of them can either be synergistic or it can be completely opposite depending upon what is the context. So just to to clearly distinguish these two, I wanted to bring it back here that DNA methylation and histone methylation are completely different, and they're not interdependent in most contexts. Apart from this, the last topic that we have for today is on histone variants. So, what is the idea behind this? What we have learned till now for today was about, okay, yeah, also keep in mind that I want to give a little more detail about histone modifications, and a little more detail about the mechanisms that are operational and how the type of modification can actually manifest in different ways for different cells. And the while some of them are hazardous, a lot of them don't really make much of a difference for the cell. We'll discuss all of that in the next class. But for today, I wanted to end on this note discussing on histone variants. So in higher eukaryotes, histone genes are present in multiple copies, and usually genes for 5 different histones are present in a cluster, which is repeated many times, and their expression is the highest during S phase. Why? Because during the synthesis phase, DNA is getting duplicated, and that DNA needs to be packaged during the remaining cell cycle. And only very residual histone gene expression occurs in the rest of the cell cycle, providing sufficient histones to cope with the demands of DNA repair and associated chromatin remodeling. Most copies of the genes which encode for any given histone are the same. However, for all histones, apart from histone H4, which means that for H2A, H2B, and for histone H3, there are some variants. What does this variant mean? It means that there is some difference from the canonical histone or the core histones with differences in amino acid sequence. In some cases, these differences are quite subtle, comprising some amino acid substitution that doesn't really manifest into anything. It cannot be considered as a histone variant. It can simply be an outlier. It can simply be a single nucleotide or single base change. For example, there are three H3 variants in mammals: H3.1, H3.2, and H3.3. H3.2 differs from H3.1 by a single amino acid substitution, which is from serine, or which results in a conversion from serine to cysteine at position 36. Now, keep in mind, while this doesn't sound like too much, the properties of these two amino acids is quite different. So, serine, while it carries the OH group, cysteine, on the other hand, carries the sulfhydryl group. And also, if there's another cysteine somewhere present, they can form that it can undergo reduction and they can form cysteine bridges, so that can affect the protein folding and the protein structure. So these are some variants which can have significant differences from the canonical histones. um, yeah, and while H3.3 has two additional substitutions, whereas H3.1 and 3.2 are synthesized only during S phase, 3.3 is synthesized throughout the cell cycle. So the variants are not equivalent in all respects, and they are actually not always hazardous also. They are sometimes required, because sometimes you need the exchange of one histone variant to another. We'll see some examples about this also in the next class. But basically, think of it in this manner that sometimes DNA in some very core regions of the or some very core chromatin needs to be opened up, especially during the process of replication. And at that point, completely unwinding the DNA, while it might not be energetically very feasible, what is more practical is to loosen up the packaging of the DNA in those local sites. And that is facilitated by substituting the canonical histone with a histone variant. That variant binds less strongly to DNA and opens up the DNA and allows replication to proceed. And sometimes the exact opposite can also happen, that it is binding DNA a lot more tightly so that DNA damage can be inhibited. So these are just some examples off the top of my head. One example here is, yeah, differences in primary structure between variants is also radical in some cases. For example, the human H2A variant, H2AZ, differs from the common variant H2A1 by 15 amino acid substitutions. That is immense. Um, in three helical domains, along with numerous substitutions, insertions, and deletions in the N-terminal and C-terminal and loop domains. So this is the H2AZ. This is H2A, the canonical histone. This is H2AZ, which has a large number of changes, and these are the different sites at which these amino acid substitutions have taken place. And the third is macro H2A, differs to an even greater extent, and it has over 200 additional amino acids added to its C-terminal tail, and it shows it associates specifically with the inactive X chromosome in female mammals, suggesting a role in chromatin condensation, which is extremely crucial to ensure that no random replication or no random expression can take, not replication, no random transcription and translation can take place from these regions. And one of the female chromosomes, X chromosomes, should be completely inactive and completely heterochromatinized. Now, there are also other contributors to this process. There is the XIST mRNA, and that is, of course, allowing the complete spread of heterochromatinization along the length of the chromosome. But this is also another process which helps in enhancing chromosome condensation and gene silencing for the female X chromosome to make it transcriptionally silent. So these were the topics that I wanted to cover for today's class. If there are any doubts, please let me know, and then we will end today's class, and we'll meet again next week on Wednesday for our next class, where we'll go into more detail about epigenetics, and then we'll go back on track and resume these topics for analysis of high throughput sequencing data and so on and so forth. Also, we are in week 7 right now. So week 8 will be almost 1/3 of the complete course. So I will be uploading all of the details. You can read up on it. And I'll also try to prepare like a small questionnaire if possible and upload it for you guys to address it. And yeah, that is pretty much it from my side. Any doubts you have, please let me know. Then we can end today's class.
Ma'am, there is a very basic question that you just mentioned, like in like 2 3 slides earlier. You mentioned that
Mhm.
in DNA methylation and histone methylation, which of the methylation leads to the like alteration of protein? Like what I can think is, uh, the DNA methylation will leads to the epigenetic like difference. Is Am I correct?
No, no, both of them are epigenetic modifications and both of them are important. But okay, see, what your question was about protein affecting proteins, right? That is different. So, um, how do I explain it? Uh, I will go into details about it in the next class, but think of it in this manner that DNA, what we are referring to here, look, okay, yeah, let's just look at this image. Here you're looking at DNA, which is completely linear. right? What we are referring to methylation here is for histone, which is bound to DNA. So this is packaged DNA, and within that packaged DNA, the histone's tail is undergoing modification. Here, there's no question of the protein. It is about how accessible that particular region of the DNA actually is to the transcriptional machinery. Now, whether or not there is a single methyl, double methyl, or triple methyl, all of that can influence whether transcription will be switched on or switched off, or somewhat down regulated or somewhat up regulated. All of those changes can take place. In contrast, when we talk about DNA methylation here, what we are talking about is hyper or hypomethylation, which means that there are certain stretches within the DNA sequence which are rich in CG repeats or CPG islands basically, that have a higher propensity to have the recruitment of the methyl group directly on the C and G residues present within the DNA. The place at which methylation is occurring is different completely between the two. The downstream effect of that methylation being present or absent can affect transcription directly or indirectly. But what the effect will be can be completely different. I cannot give you a very clear-cut answer as to whether or not a protein will be switched on or switched off because it is very context specific. Like for example, here if you have the CPG island, which is very upstream of a tumor suppressive gene, when that is undergoing hypermethylation, then the tumor suppressor expression is shut down or is lost or is repressed, and that is helping cancer. Similarly, when there is hypomethylation or less methylation of an oncogene, that is leading to cancer as well. But in this case, the presence of modification on the histone can have very different effects, which we'll learn about in the next class. And whether it is present very close to the DNA which is surrounding a transcriptional start site, whether it is one methyl or dimethyl, all of that can influence it. So net conclusion is this, that depending upon the context and the region, both of them can affect transcription and eventually the translation, and both of them can have positive or negative effects on disease progression. But yeah, and I know it's a little confusing, but it's, think of it as multiple levels of control. Both of these are epigenetic controls, but the control is executed through different molecules. One is protein, one is DNA. Was I able to explain it somewhat?
Yes, yes, ma'am.
Yeah, okay, but yeah, we'll learn more about it and how both of them actually interact with each other also, DNA methylation and histone. Um, Whether or not they can actually co-occur or one regulates another, all of that we'll learn about it. So that's why I've kept it as like a separate topic also within the course handout. So yeah, any other queries?
No, ma'am.
Yeah, this is what we'll pick up in the next class. But for now, we can end today's class. Thank you very much for joining. I'll share the details over email and then we can have the discussion. Okay, thank you. Have a good night. I'll end the class now.